Welcome to Lab Lecture 14, where we're deep in the heart of identifying unknown bacteria using physiological and biochemical testing. And today we are going to cover so many exciting different biochemical tests and review what you saw on your throat swabs from last time when you streaked from someone else's throat onto a BAP, a blood agar plate. So as you're looking at those, when you get to lab today, you're going to get to answer the question of what 3 types of hemolysis can you observe on this BAP plate. And you'll notice in this picture, I've got some rad pictures I've accumulated over the years, that over on the left is a type of hemolysis in which you can't see through the zones. In fact, the zones look more opaque than they did before they formed. That is, you can see less through them. Whereas in the middle, those zones look like you can see straight through them. And then on the far right, those zones look like you can't see any kind of zone at all. That is to say, there is no zone. So these are the 3 different types of hemolysis that you might see on a blood agar plate. So for your pre-lab today, I want you to write down in order of what you see there, what type of hemolysis, what's the first one, the second one, and the third one. So when you look at your throat swabs today, you're going to see each type of hemolysis potentially. Some of you will only see alpha and gamma hemolysis, and occasionally some of you will also see beta. About 15% of people harbor Staphylococcus aureus in their nasopharyngeal area, and remember that that is a beta hemolytic bacterium. So you are normal if you're amongst those 15% of people. You're just unique, and it doesn't mean that you're sick. Now, on the other hand, if you see Streptococcus pyogenes on your plate, another beta hemolytic bacterium, it either means that you have strep throat or it means that you're a carrier for strep throat. So the clear zones formed by Streptococcus pyogenes are the indicative ones of strep throat carriers or strep throat sufferers. Now, we tend to see an awful lot of alpha hemolysis on our throat swabs. A lot of our normal florobacteria, like Streptococcus mitis, are alpha hemolytic. So let's look at a throat swab so that we can see what we might expect. And everybody looks at these the very first time they see a throat swab and they think, oh my God, that's disgusting. Not at all. It's just your normal flora, your healthy microbiota. What would be disgusting is if you saw a monoculture in your throat. That is, if you saw only one bacterium and it was all beta hemolytic, then you would be like, oh no, I have a disease, right? There's a lack of biodiversity, and this particular microorganism is lysing the heck out of red blood cells. So it would be indicative that you had strep throat. So these throat swabs are from students who have normal flora in their throats. Their microbiome looks very healthy in their nasopharyngeal area. And we're seeing a lot of alpha hemolysis. Remember, that is the green kind of opaque looking zones. We're also seeing a little bit of gamma hemolysis. And in fact, there are some little colonies here where you can't see any type of zone around them at all. And in fact, even the larger colonies look like they're not showing much of a zone. Of course, you would also want to hold this throat swab up to the ceiling so that you could see through the light whether or not your organisms are forming any kinds of zones of hemolysis. And occasionally, as I said, 15% of you will see that you have beta hemolysis, the very, very clear zones. So we would want to be able to, if we saw those beta hemolytic zones and we thought someone had strep throat, we would want to be able to isolate that bacterium and then put it, so we would streak and re-streak, right? And we would put that onto a new plate and we would try to determine what kind of antibiotic that organism was sensitive to. So remember doing the Kirby-Bauer assay in a couple of labs ago, that was helpful in determining the therapeutic strategies that we might put forward to treat these organisms. Also recall that on the throat swab plates, you not only streaked them, but you also stabbed them. So after streaking into half of the plate, you kept a certain amount of culture on your loop, and then you stabbed deep down into the agar. So this particular image shows the base of those stab marks for a plate that was streaked with Streptococcus pyogenes. Remember, the strep throat causing bacterium. Streptococcus pyogenes produces a type of hemolysin that lyses red blood cells only in anaerobic conditions. And so it's only visible at the very, very bottom of the stab mark. So here you can see these very, very, very clear zones of lysis that occur at the bottom of the stab mark. These are indicative of strep throat. They're indicative of Streptococcus pyogenes. So when we did our throat swab plates, we did the streak stab technique so that we could try to find this particular hemolysin that's inactivated by oxygen. It's called Streptolysin O. So Streptolysin O is only visible at the very bottom of the stab mark. Now, on the other hand, you also noted in that image that there was a lot of beta hemolysis around the individual colonies. Let's look at that again. So notice that this is also beta, very see-through. You can look through the whole darn thing. That beta hemolysis is Streptolysin S. So there are two streptolysins produced by Streptococcus pyogenes. One, Streptolysin S, S for stable in the presence of oxygen, and Streptolysin O, O for inactivated by oxygen. So when we streak a plate with the strep throat causing bacterium, we can find both Streptolysin S on the surface of the plate and Streptolysin O deep down in the stab marks. So if you did happen to see a lot of beta hemolysis on your throat swab plate, then you would want to look also at the bottom of your stab marks to see if they become even more clear at the very base of the stab mark, and that would be indicative of Streptolysin O. So regarding the staphs, we need to change our attention away from the streps and towards the staphs, but let's use a natural segue. So say that you think you do have strep throat. You saw a bunch of beta hemolysis on the plate. You see the really clear zones at the bottom of the stab mark, in which case you might think, gosh, I should gargle with some salt water, right? Because salt is very suppressive against the streps, but it allows the staphs to grow. So that helps us with this first question of which organism, Staph aureus or Streptococcus mitis, would grow on an MSA plate. Well, we know that the staphs will grow in the presence of salt, whereas the streps will be killed by that salt. So Staphylococcus aureus will be the one to grow on that plate. Now, further, we remember that MSA stands for salt agar, but it also stands for mannitol salt agar. So not only is it a selective agar that suppresses the growth of streps, but it is also a differential agar that differentiates based on the ability to ferment mannitol. And my cat Gabby wants to meet all of you right now, so she's looking for a little bit of a photo opportunity. So when we look at an MSA plate, we see something that looks like this, where the color change that occurs if mannitol is fermented is a yellow color. So notice that over on the left-hand side of the plate, where Staphylococcus aureus is growing, it's also turning the plate yellow. And that yellow color is indicative of the mannitol being fermented. So mannitol fermentation is causing an acidic byproduct that is turning the pH indicator from red to yellow, and then we're seeing that as well. So that's a nice little bit of review. Let's turn our attention to the other way that we differentiate staphs and streps. So one would be salt tolerance, the ability to grow on a mannitol salt agar, but the even more quintessential test that allows us to differentiate staphs and streps is the Catalase test. So the Catalase test looks at the ability to create those magic bubbles upon being added to hydrogen peroxide on a glass slide. Remember, we did this one in lecture. So notice that if an organism can detoxify hydrogen peroxide, it converts it into water and oxygen gas. And you'll notice that this slide has 2 drops of hydrogen peroxide, and one of them has catalase. You can see that it has bubbled like crazy, and that one is catalase positive, whereas the other one does not have any catalase, and it has stayed without bubbles. This is the most quintessential test for differentiating staphs from streps. Now, remember, we talked about it in lecture, and what we said about it was that, in fact, the staphs are one of those organisms that live in an oxygen-rich environment. So the staphs, of course, are going to have to detoxify a certain amount of naturally occurring toxic derivatives of oxygen. H2O2, or hydrogen peroxide, occurs in small amounts in any oxygen-filled environment. So the staphs that live on your body surfaces have to be able to detoxify it. They need to be able to take the naturally occurring hydrogen peroxide and convert it into water and oxygen gas. So we can write down the answer to these questions, and in fact, this is your pre-lab number 2. Which members of which 2 genera are catalase positive, and members of which 2 genera are catalase negative? So why is this such an important test? To allow us to distinguish these 4 genera into their categories as either catalase positive or catalase negative. Sweet. So moving along, let's talk about a very phenomenal test called the Coagulase test. So say that you have received an unknown bacterium, or maybe you took an unknown bacterium off of somebody's throat swab, and you're isolated that unknown bacterium. And now you want to know, is it gram positive or is it gram negative? You do a gram stain on it. Okay, you find out it's gram positive. So from there, you think, I'm going to do a catalase test, right? So you do that catalase test and you see the magic bubbles. You're like, wow, I think I have a staph. Maybe I have Staphylococcus aureus. So at which point, you potentially might want to go ahead and do an MSA plate to see if it ferments mannitol. But the really cliche test that you're going to want to make sure you do is a coagulase test. 97% of all staphs are coagulase positive. So going ahead and putting your unknown bacterium into rabbit plasma, you'll see whether or not it can turn fibrin into fibrinogen and lead to that clot. So a coagulase positive will do so, and you can see in the picture that I took here that a coagulase positive organism causes the plasma to stick in the bottom of the tube. And in fact, you can even turn it sideways and upside down, and if the clot is firm, it will stay in the bottom of the tube. So Staphylococcus aureus is coagulase positive, whereas other staphs are coagulase negative. For example, Staphylococcus epidermidis was placed into this tube, and it is clearly coagulase negative. This is a really definitive test to allow you to identify Staphylococcus aureus, and it's not the only definitive test that we're going to talk about today. The streps also have definitive tests, and the Taxos tests are those. Taxos P is a disc that is infused with the antibiotic Optichin. And Optichin is going to be very effective against Streptococcus pneumoniae. The causative agent of upwards of 95% of all cases of pneumonia is Streptococcus pneumoniae. So Streptococcus pneumoniae is Optichin sensitive. So if you put it in a ring around a Taxos P disc, it will show a lot of inhibition in its growth. Whereas like our normal flora streps that just are natural in our throats, they aren't killed by Optichin. So notice in this picture, this is one of our normal flora streps. It does not show any zone of inhibition. And although this isn't the best picture I've ever taken, you can notice a big zone of inhibition around the Taxos P disc here, and know for sure that that must be Streptococcus pneumoniae. Now, I have a sweet mnemonic for helping to remember this. I'm going to write it down and share it with you. That is, Streptococcus pneumoniae is sensitive to Taxos P. Now, the other type of Taxos that we'll be testing with is Taxos A. And Taxos A has bacitracin, and it is definitive for it tests amongst the beta hemolytic streps, but explicitly Streptococcus pyogenes is sensitive. So Taxos A, bacitracin, differentiates beta hemolytic streps and specifically Streptococcus pyogenes, the causative agent of strep throat, is sensitive to Taxos A. So we can look at this plate, and this is your next pre-lab question. Is this organism capable of causing strep throat? So if you think it is, go ahead and write a little note on your third pre-lab question. So Taxos A disc inoculated with bacitracin, and it differentiates amongst beta hemolytic streps, largely telling us or allowing us to know definitively whether we have Streptococcus pyogenes, the strep throat group A, hemolytic, beta hemolytic strep. Beautiful. So we have a lot of sweet differential tests that allow us to, in some cases, clearly define or be presumptive for certain bacteria. So we have certain definitive tests. This is another one of those for gram-positives, the Bile esculin agar, and it is particularly good at allowing us to identify what I think of as the duck out of water, and this is Enterococcus. So remember how we've talked a lot about how gram-positives tend to find their home in our nasal, pharyngeal, and skin areas, whereas gram-negatives tend to be more at home in our gut. So with the Enterococcus, defined by or found using the Bile esculin agar, it's kind of weird because it's a gram-positive, but it likes to chill out with the bacteria that live in your gut. So that's why I say it's a duck out of water, is because it's a gram-positive that likes to chill out and hang with the gram-negatives down in your gut, which must mean that it's resistant to bile. It must also mean that it's able to metabolize in the presence of bile. So we use a Bile esculin agar to find it and to determine that that's what we have found. So this agar is both selective and differential, and it really does allow for the growth of only the Enterococcus species, both faecium and fecalis. So bile inhibits the growth of gram-positives, sodium azide inhibits the growth of most gram-negatives, leaving us then with just this crazy bile-resistant Enterococcus. And it metabolizes, of course, in the presence of bile, so it's able to take this esculin, converting into esculitin, which then turns the medium black. So we'll look at a picture where we can see two that are negative, or I should say one that's uninoculated and one that's negative, and then notice that Enterococcus fecalis turns this entirely black, showing us that esculin has been metabolized in the presence of bile. And we can say, hey, you, B, right there, you are Enterococcus fecalis. Sweet. So as we finish our coverage of gram-positives and some of the awesome tests that we use for those, we can transition into looking at some of the biochemical tests that we use for gram-negative bacilli. One of the most beautiful agars that we work with for gram-negative bacilli, I think, is MacConkey agar. MacConkey is also both selective and differential, so it selects against gram-positives and it selects for gram-negatives. So it's kind of, looking to do something opposite of the Bile esculin agar. This time we want the gram-negatives to grow and we want the gram-positives to die, and we use bile to do that. And of course we know, hey, Enterococcus could live through that, right? So it doesn't get rid of all the gram-positive that way. But there are some other selective ingredients in there, namely dyes, but we also allow the differential nature of this medium by putting in a pH indicator that turns from a kind of colorless sort of purpley into a really bright pinky red, I say Barbie pink color. So you can see here that this organism, this is a plate streaked with E. coli that is a quintessential lactose fermenter. And so we recognize that it's turned all of the agar this bright pink, and the colonies have turned bright pink. So it's a really definitive positive for lactose fermentation. Now, if you want to know what a negative looks like to compare it to, this is a negative. Really kind of that nondescript purpley color, no bright Barbie pink like this one. Oh, so pretty, right? Compared to the negative that really stays that nondescript kind of, purpley color. So we can turn our attention then from MacConkey agar, which looks at lactose fermentation with our gram-negatives, to glucose tubes that look at glucose fermentation, of course. And we know that the glucose tubes also have a pH indicator in them, but in this case, it turns from a kind of reddy color to a yellow color. So this actually is a pH indicator. I'm going to jot down what it is so that you can have in mind this particular unique pH indicator. It is called phenol red. So phenol red turns from red to yellow when there's acidic fermentation. So if glucose is fermented to acid byproducts, as we see in so many fermentations, the phenol red will turn from red to yellow. So we can recognize the capability of fermenting glucose. But this tube does not only do fermentation. So if this is our glucose broth tube here, it begins off with a reddish color and that will turn to yellow if there is fermentation. So if it's positive for glucose fermentation, we'll get that conversion to yellow color. But not only that, we also see inside of this tube an upside down little tiny tube that sits inside of the larger tube. Now this is all filled with liquid as well. So the broth goes all the way up into this little teeny tiny tube. If the bacteria, after fermenting to form an acidic byproduct, convert that acid, which often is formic acid, into gas, carbon dioxide gas and hydrogen gas, then those two gases get caught in this Durham tube. And when we see the formation of the yellow color, we also get a Durham tube that has a big bubble in the top of it. So this whole area up top is gas. And then the liquid is down here, but it's not in the gassy part. So there's a big bubble in the Durham tube. So CO2 and hydrogen gas form that bubble. So this is a recognition that many members of the family Enterobacteriaceae, which includes E. coli, Yersinia pestis, Shigella dysenteriae, and others, convert pyruvate formic acid and then to hydrogen and carbon dioxide. We get the formation of the CO2 and H2. And that actually allows us to differentiate amongst those that do this versus those that don't. So E. coli makes a lot of gas. It is a super gassy bacterium, whereas Shigella dysenteriae is not gassy. So even though we can see fermentation of glucose in some more pathogenic organisms, we will not see the formation of the gas. And that can be a very distinguishing feature for differentiating Enterobacteriaceae. Let's look at a quick picture of this so we get the feel for what it really looks up close and personal. Here is that gassy bubble that's formed in the Durham tube. We additionally notice that the phenol red has turned from red to yellow, so this is definitely fermented glucose. Beautiful. Let's turn our attention then to the SIM tube, which is a test that we've already talked about, so we can review it a little bit. So what will be the appearance of a SIM tube that is inoculated with an organism that produces hydrogen sulfide gas? Remember that hydrogen sulfide can be the product of desulfurylating cysteine. You'll recall that cysteine is that sweet amino acid that has an R group. So if we remember back to our amino acids, let's make this a more visible color. So if we remember back to our amino acids with their central carbon and their hydrogen, their amino, and their carboxyl, as well as their R, well, cysteine is the one that has the sulfhydryl in it, so it's an S bond H. This can be desulfurylated, and that leads to the formation of hydrogen sulfide. That's one way that we can see the production of H2S. But of course, you all can tell me what the other way is. If we think about those bacteria that respire anaerobically and they use sulfur or sulfate as their terminal electron acceptor, then they produce the smelly rotten egg hydrogen sulfide gas, right? So then the question is, how do you know? So for your next pre-lab question, number 4, tell me which one of these organisms is capable of producing hydrogen sulfide gas. So we can say that, and additionally add to that question, what are the 2 other things detected by a SIM tube? Rockin'. Finally, we get to the medium of the hour, the Kligler's iron agar, or KIA. This is one of the most complicated forms of biochemical testing that we will talk about. So I want you to recognize that KIA is going to basically be looking for the ability of an organism to ferment both glucose and lactose. We want to do it all in one fell swoop. So if anything, KIA is a multitasker that doesn't realize it's not a good multitasker. We know a lot of people like that, right? So KIA tries to do a ton of things at once, but then it's not very, very good at it. So keeping that in mind, what it's trying to do is it's trying to look for both glucose and lactose fermentation at once. And the way that we do this is we put lactose into the tube at super high concentration. We put glucose into the tube at lower concentration. So a bacterium that can ferment glucose is going to chow down on that glucose first. It's like the preferred growth substrate. It's like if you go to a buffet, it's the thing on the buffet line that gets used up first. So all of the bacteria that can use glucose are going to go for that glucose first. They're going to eat it entirely, and then they're going to, if they can, they're going to shift and they're going to use lactose. But within a KIA tube, we additionally see that it contains phenol red, except for this time it's in an agar slant. So we've got this phenol red present in agar, and so it begins off red. Now, if we put a glucose fermenting bacterium onto this KIA, then it's going to ferment the glucose, and you guessed it, it's going to turn that phenol red from red to yellow. So the entire agar is going to become yellow. And we can say, okay, that organism fermented glucose. But this is going to happen at some point in the middle of the night while we're at home sleeping, and the bacteria are gradually running out of glucose. So they're going to run out of glucose. So we can say that at this point, glucose fermentation has taken place. Glucose fermented. Now, if we let it sit longer, though, the bacteria are going to have to make a choice. They're going to have to move on to lactose. So say that a bacterium can ferment lactose. Ooh, that's the fun arrow of the day. So if they can ferment lactose, then lactose will be fermented. And you guessed it, the slant will stay acidic and it will stay yellow. So in the next morning, when we all come in and we look at this KIA tube, we can say to ourselves, wow, that bacterium fermented glucose, it used it all up, and then it moved on and it fermented lactose. So we can say that a yellow slant over a yellow butt equals both glucose and lactose fermentation. Yellow over yellow means both have been fermented. Now, let's alternatively say that this bacterium runs out of glucose in the middle of the night and it says, okay, I got to move on to lactose, but it can't. Let's say that this is a bacterium that cannot ferment lactose. So in which case it has to move to another source of energy, and instead it ferments amino acids. So it actually utilizes, I'm going to just use shorthand, AA metabolism. Amino acids are going to be metabolized. So AA metab. Now we know that amino acids, as we drew in the prior picture, let's do it again, because let's face it, you can actually never really get enough of drawing amino acids. I mean, if you just want to spend all your free time drawing amino acids, not only is it fun, but it is also educational. So the amino acids, when they get broken down and used as an energy source, they have to be deaminated. So this leads to the formation of ammonia or ammonium in the medium. And that's basic, right? This is so base, right? So basic ammonia is going to convert the yellow back to red. But also remember that amino acids have to enter into the TCA cycle, so they do so aerobically. So that means that the slant of the surface of this agar tube is the one that's going to turn red. So when you come in the next morning, you're going to see a red slant where the amino acids were utilized over a yellow bud, because the amino acids can only be used aerobically and not anaerobically. So an organism that is red over yellow can ferment glucose, but it cannot ferment lactose, red over yellow. Okay. But what about the poor guys out there, maybe not so poor, that don't ferment either lactose or glucose? Maybe something like Pseudomonas aeruginosa, one of my favorites, doesn't ferment at all. It's like, dude, I am all aerobic all the time. Pseudomonas aeruginosa doesn't ferment. So it's going to, as you might guess, leave this tube red over red. It's simply not caring about either one of those sources. So red over red means that neither one is fermented. Sweet. So I hope you're starting to understand KIA. Let's look at a couple of examples as we move along. So check it out. If a KIA looks like this, yellow over yellow, we know that it ferments both glucose and lactose. Red over yellow, we know that that means the amino acids got broken down, they raised the pH, particularly in the aerobic environments. Check that slant, a beautiful red slant over a yellow butt means that we have seen fermentation of glucose, no lactose. And of course, red over red, as we'll look at next, is going to mean that neither was fermented. So a red slant over a red butt likely means that organism is non-fermentative, like Pseudomonas aeruginosa. It likes to use the ETC, and it likes to use oxygen as its terminal electron acceptor, and it likes to live aerobically all the time, and it doesn't like to ferment. It's not its style, right? It likes to live aerobically, not fermentatively. Now, this last one is a bit perplexing, and I have a question for you, and in fact, you can add this to your pre-lab. So for this one, we've got a clear, obvious formation of black precipitate. So remember how I said that KIA likes to do too many things and it doesn't really in the end do them all that well? So KIA also looks for hydrogen sulfide production. So notice in here, if you look there, you can see some H2S, you see the black precipitate. So you know that desulfurylation of cysteine or that production of H2S via anaerobic respiration has occurred, and that has accumulated hydrogen sulfide gas. But looking at this tube, what can you tell me? Can it ferment glucose and lactose, this bacterium? Yes, it does produce hydrogen sulfide, but can it ferment glucose? Can it ferment lactose? You tell me on your pre-lab. Sweet. So this is a cool picture where you can see yellow over yellow, red over yellow. You can see the H2S production, and then the all aerobic all the time Pseudomonas aeruginosa, where it's not using or fermenting either glucose or lactose. Cool. Our next gram-negative test is the oxidase test. This is the most important test for gram-negatives. This is the most important test for gram-negatives. Wake up if you're not awake. We need to be able to do the oxidase test on our unknown. And in order to do this effectively on your unknown, you need to watch Virtual Edge. You need to watch Virtual Edge to make sure that you know how to do this test, because it is a time-dependent test. So you're going to take your bacteria off of the slant, you're going to put them onto the Whatman filter paper, and you're going to put this chromatogenic dye on it. And you're going to look to see which organisms have the terminal oxidoreductase called cytochrome C oxidase, aka complex 4 of the mitochondrial ETC. Some bacteria have it, some bacteria don't. We're looking for it. And if bacteria have it, then they convert this chromatogenic dye to a dark purple color quickly. That is the key. They do it quickly. So if you sit around and lollygag, every bacterium is going to turn purple. But it's the immediate turning of purple upon adding this dye that tells us that an organism is oxidase positive, aka it has cytochrome C oxidase complex 4 of the mitochondrial ETC. So when we look at this picture, we have to say that this is immediately after adding the reagent, and then we can determine that B is the one that is oxidase positive. That is, B is Pseudomonas aeruginosa. Pseudomonas aeruginosa and Neisseria are oxidase positive. We don't work with Neisseria, so guess what? If your unknown bacterium turns oxidase positive, you can be pretty dang sure you're in the genus Pseudomonas. So this is a very important test for your gram-negative unknowns, because if it's negative, then you'll be thinking, I might be in the family Enterobacteriaceae, Shigella, E. coli. Hopefully not, but Yersinia pestis, right? So these are some good examples of things that are not oxidase positive, where Pseudomonas is oxidase positive. We're going to end today looking at the Urease test. And I have to tell you, if I'm not near the test I love the test I'm near. But I'll be honest, I think the Urease test is absolutely my favorite of the biochemical tests. Because let's face it, what could be more beautiful than this bright Barbie pink Urease test? Oh, it's so gorgeous. Okay, so let's recognize that Urease is produced by certain bacteria, and it splits urea into ammonia and CO2. Now, you have smelled this. Have you ever gone to change your kitty litter box and smelled like ammonia? Yep, that's urease. So urease, and likewise, some of you work in healthcare. Have you ever worked with a patient who had a urinary tract infection and it smelled like ammonia? Yep, that's the bacterium that is converting urea. So urea into ammonia and CO2. So this is the enzyme urease. Now, that equation is totes not balanced, so go ahead and add that to your pre-lab. Balance this equation for me and just try on your chemistry skills. Who says they never apply in the biological sciences? You bet they do. So recognize then that Proteus mirabilis, remember the swarmingly motile bacterium that we've been working with? Of course, it's swarmingly motile. It can really get around in the urinary tract, and it does. So Proteus is the causative agent of UTIs, particularly in pregnant women. It is one that can most certainly be associated with ammonia-smelling urinary tract infection. So this is cool because guess what? It turns out that once again, the pH indicator in this tube is, yes, you guessed it, phenol red. And of course, when ammonia is formed as a byproduct, this is basic, so it turns the organism bright Barbie pink. What a wonderful place to end the day.